Good morning, children. Sairam, welcome to this session. Today we are going to start with a new chapter, lesson number 8, Cell Biology and Biotechnology. Okay, so we have finished with the 9th lesson. Now I am going back to the 8th lesson. The name is Cell Biology and Biotechnology. Okay, in this lesson we'll see what we are going to learn. We are going to learn about cytology or cell biology. We are going to learn about stem cells, biotechnology and its applications, and important stages in development of agriculture. All these related to biotechnology. Last year in 9th standard itself we had learnt about tissue culture, isn't it, children? Hope you remember what is tissue culture. Okay, that you can just revise before learning this lesson, but no questions will come from 9th standard portion. But about the stem cells we have learnt in plants. Okay, 9th standard. Now we will learn about what is cell biology or cytology. Okay, so we have learnt about structure of cells, cell organelles. All this study of cells is called as cell biology or cytology. So what is cytology? The study of cells is called as cell structure, cell organelles. Okay, and cell division, all these comes under cell biology or cytology. Okay, now we'll see what is the remaining. Now what is meant by stem cells? So these are special types of cells present in the body of multicellular organisms. There are some cells which are called as stem cells, and these give rise to all other types of cells present in the body of multicellular organism. So the main type of cells present in the multicellular organism from stem cells. From these different types of cell arises. Similarly, these cells play an important role in wound healing. We learnt in mitosis. Okay, so there is multiplication of the stem cells in wound healing also. Okay, so this is the special type of cells present in the body of multicellular organisms, and they can give rise to all other types of cells present in the body. Okay, they are called as stem cells. We have learnt about stem cells in the plants. Okay, in the 9th standard. Now we shall study about the stem cells in animals, particularly in human beings. Now a new organism is formed from the zygote, that is the union of male and female gamete. This we all know. A new organism is formed from the fusion of the male and female gametes. At the earliest stage of development, organism is in the form of a mass of cells. So in the initial stage, when there is fusion of male and female gametes, there is a mass of cell formed. Okay, all the cells in that mass will be alike. So those cells are called as the stem cells. So what is stem cells? Now new organism is formed from the zygote. You all know this. From how is the zygote formed? From the fusion of male and female gamete. At the earliest stage of the development, the organism is in the form of the cells are in form of a mass. This mass of cells, all the cells in that mass are looking alike. So these cells are called as the stem cells. Understood? Now, during further development, these cells form any type of cell, different types of tissues, and perform different functions in the body. So after development, they form different types of cells, tissues, and all these different, and even organs. So this is called as the differentiation of stem cells. Okay, so this is in the initial stage, in the embryonic stage. Initially, when the zygote is formed, they are in the form of a mass, and all the cells in that mass will be similar, and that is called as the stem cells. Understood? Okay. Now this stem cells can be preserved also. Okay, this is given in the box inside in the textbook. You can just go through. For the purpose of preservation, stem cell samples are carefully collected from the sources of cord blood, that is from the umbilical cord. When it is connected to the placenta, okay, that cord blood, red bone marrow of embryo, that is the or red bone marrow, or the embryo, that is the blastocyst, that means the earliest stage, and are kept in the small sterile vials. Vials means a small glass bottle or container where we can store it in a preserved condition. Okay, these vials are kept in liquid nitrogen at minus 1350 degrees Celsius to 1000 minus 1, is stored 1900 degrees Celsius. Okay, so this is for the preservation. They can be, this stem cells can be taken from the cord or red bone marrow or the blastocyst on the early embryonic stage, and they kept in the small sterile vials. Okay, and they can be stored in liquid nitrogen. So this, like this, the stem cells can be preferred, and different types of other organisms or other organs can be formed from this. Okay, now stem cell research. In biotechnology, stem cell research is a revolutionary event after cloning. Cloning means forming the replica, same to same is called as, or even the DNA structure, DNA, everything will be same in cloning. So after this cloning, there is this stem cell research is a revolutionary event. This technique has the potential of bringing about fundamental changes in the medical science. Depending upon source, stem cells are of two types. There are two types of stem cells. One is the embryonic stem cells and the adult stem cells. There are two types. One is the embryonic, another one is the adult stem cells. What is meant by embryonic stem cells? Division of the zygote starts and thereby it is converted into embryo. This we all know. Once there is union of male and female gametes, there is the zygote form. And that is converted into embryo, isn't it? Cells of the embryo undergo repeated mitotic division. So there is repeated mitotic division in the embryo, and this there is cell differentiation. Okay, and that starts from the 14th day of conception. So once there is fusion of male and female gametes, that is the embryo form. So once there is slowly differentiation starts from the 14th day of conception. Okay, then cells of the different organs like osteocytes, that is the bone cells, hepatocytes, liver cells, and neurons are formed due to differentiation. So from the stem cells, what of us form? Bone cells are formed, liver cells are formed, and the neurons are formed. From when? From the 14th day of conception. So once the zygote is formed, there will be division, multiplication of the cells. And first, what all cells will be formed? Osteocytes, that means bone cells, hepatocytes, that means the liver cells, and the neurons are formed due to differentiation. Then embryonic cells before differentiation are called as embryonic stem cells. So what are embryonic stem cells? Before differentiation, this embryonic cells are called as embryonic stem cells. Okay, from this all other, you can see the diagram also. I'll show you. Now there are 220 different types of cells in the human body formed from single type of cells. So how many type of human cells are formed? 220 different types of cells are formed. Human body are formed from the single type of cell, that is from the embryonic stem cells. Thus, stem cells are primary type of undifferentiated cells with self-multiplying ability, and they are parent cells of all types of human cells. So which is the parent cells? We learned in 9th standard also. From this all the cells will be formed. So same way even in the human also, this embryonic stem cells, from this or 200 different types of cells can be formed from of the human body. This property of stem cells is called as pluripotency. What is meant by pluripotency if it comes? So with the multiplication of cells, 220 different types of cells are formed from one type of single type of cells, which is called as the embryonic stem cells. This property is called as pluripotency. It has been found that if these stem cells are collected well before the beginning of differentiation, of that is on 14th day, so only on the 14th day there will be differentiation. So before differentiation, that is on the 5th to 7th day, if they are collected and cultured with different biochemical stimulus in laboratory, that is from which day? Before the conception, before the differentiation starts when after 14th day. So before that, if we collected from 5th to 7th day and it is cultured with certain biochemical stimulus in laboratory, and as per the stimulus we can transform themselves into desired type of cells, thereby tissues, and finally into organs. So before 14th day, if you take that and and the biochemical stimulus is given to that, and we can transform this stem cells into whichever tissues or organs. You can see here organs as per our desire. That is when from the 5th to 7th day before the conception takes place. You can see here. So in between the stem cells, you can see the neuron, bone cells, all the cells will be formed. Okay, liver cells, and from there the other organs like brain, intestine, liver, heart, all the organs can be formed from the single type of cells which is called as the stem cells. This is the embryonic stem cells. Okay. Now let us see the next type, which is called as the adult stem cells. Stem cells can be obtained from the body of adult. So first we saw in the embryonic stage. Now it is even in the adult stage also. Adult also we can find this stem cells where there are three main sources of stem cells in the body of adult person. Stem cells can be obtained from red bone marrow, adipose connective tissue, and the blood. So from where we can get the stem cells? From in the adult, from red bone marrow, adipose connective tissue, as well as from the blood. Besides, stem cells can be obtained from the cord blood immediately after birth. So immediately there is a placenta connecting between the child and the mother. So when that cord is immediately after birth, that cord will also have umbilical cord which is connecting, that also will have this adult stem cells. Understood? Okay. Now what are the uses of this stem cells? We'll see the uses. One is regenerative therapy. That means under that there are two. One is cell therapy. Stem cells are used to replace the dead cells in the case of conditions. See, sometimes in some disease like Parkinson, diabetes, many other diseases, the cells will die. Okay. So to replace this dead cells, we can use this stem cells. That is regenerative therapy. When as so many diseases, this all the cells will die. So to replace that cells, we can use this stem therapy. And to produce red blood, you produce blood cells required in conditions like anemia, thalassemia, leukemia. Sometimes in this diseases, we will require some red blood cells or blood cells. Okay, like platelets and all those white blood cells. So many are. So this can be, we can use the stem cells to produce this blood cells. Okay, and it is also very useful in organ transplantation. In the case of failure of organs like kidney, liver, and those can be produced with the help of stem cells and transplanted. So usually organ transplant is done from the human. Okay, either some can be taken when they are alive. Kidneys you can transfer when they are alive. One kidney, it will function. People can survive with one kidney, but other organs you cannot transplant. When some of the organs like heart and all, we cannot transplant when they are alive. So this so when there is failure of some organs like kidney and liver, we can use the stem cells and produce their organs, and it can be transplanted. You can see the diagrams. Okay, so what is the use of the stem cells? They undergo multiplication, isn't it? So they replicate themselves. So there are differentiation taking place, and it can be transformed into any other types of cells also. See here, you can see there are muscle cells. You can see all those skeletal, all the different muscles, and all those cells can be, tissues also can be produced with the help of stems. Okay, now we'll see about organ transplantation. Okay, this is a small topic related to the stem cells only. Various organs in the human body either becomes less efficient or completely functionless due to various reasons like aging, accidents, infection, disorder. This you all have heard about organ transplantation. Okay, due to any accidents or any reason, infection or disorder or aging, some organs becomes ineffective. So life of such person becomes difficult, or even fatality may occur under such conditions. However, if a person gets the necessary organ under such condition, his life can be saved. Okay. So if you get the organ, when you can replace that dead organ or which is inefficient, we can save the person's life. How can we do that? Availability of donor is an important requirement in organ transplant. You cannot force anybody to give their organs. Can you do that? No. So there should be donors who are willing to donate their organs. Each person has a pair of kidneys. As the process of excretion can occur with the help of single kidney, person can donate another one. So I told you, kidney, there's a pair of kidneys. So even functioning of the excretion takes place with the help of one kidney. Even with one kidney, people can survive. So we can willingly give another kidney to the person who is in need. Similarly, skin from certain parts of the body can also be donated. Sometimes some person who got burns, very terrible burns, and their outer skin is fully burned, you cannot take skin from their own body, isn't it? So we can donate even the skin from our body, and that skin, it will grow on its own for us, and it can be donated in case of surgery. Okay, so skin and even kidney can be donated when we are alive. Okay, then various factors like blood group, diseases, disorders, age, etc. of the donor and recipient need to be paid attention during transplant. See, it should match. You cannot transplant, anybody cannot give, so the blood group should match. Okay, they should check their disease. If they've got any diseases like AIDS or HIV, they cannot transplant. Okay, even there is age matter. You cannot be very old when you are donating any organ. So however, other organs cannot be donated during lifetime. Organs like liver, heart, eyes can be donated only after death, isn't it? Thus, this has led to the emergence of concept like posthumous, after death donation of body and organs. So this is posthumous, means after death. So people, when they are alive, they will write it and keep. So after death, eyes we have to be donated to somebody. Okay, to the needy person. The organs also, which can be like liver, heart, within few hours, if we take that from the dead person, and we can give it to the needed person, and they can survive. Okay, now organs and body donation. Okay, human bodies are disposed of after death as per traditional customs. Usually what they do, what any person dies, it is usually as per custom, or any custom, it is disposed of. We don't keep it for more than few hours. However, due to progress in science, it has been realized that many organs remain functional for a certain period even after death occurs under specific conditions. Even after natural death, after the person dies, even after death, some of the organs are will be functioning. Concepts like organ donation and body donation have emerged. Why is body donated, children? It can be as an example for the medical students. Okay, when we do practicals in medicine, they will open the body of a real human being. That should be a dead man, isn't it? Dead person. So they can donate their body to the hospitals, and they can do the surgery. Medical students can learn by opening the dead body of the man. So concepts like organ donation and body donation have emerged recently after realization that such organs can be used to save the life of other needful persons. A liberal view behind the concept of organ and body donation is that after death, our body should be useful to the other needful person, so that their miserable life would be comfortable. So many people, those are blind, or those who are in need, their organs are failed. So when person dies, immediately if you transplant that organs, other person will survive, and they can even see. Okay, so there they can lead a comfortable life. So once we have dead, we can donate our organs, which can be donated, which is functional for few hours even after our death. So this is organ and body donation. You can see the picture also. I'll show you. Awareness about these concepts is increasing in our country, and people are voluntarily donating their bodies. Okay, before dying itself, we have to write and keep, because it is only the with the person's wish we can do this. Life of many people can be saved by organ and body donation. Blinds can regain their vision. Life of many people can be rendered comfortable by donation of organs like liver, kidney, heart valve, skin, etc. Similarly, body can be made available for research in medical studies, as I told you. Many government and social organizations are working towards increasing the awareness about body donation. I told you before itself, we have to decide whether after death our body should be donated, either our organ should be donated, and then it can be done as per the procedure. Okay, you can see the organs which can be donated: heart, pancreas, kidney, skin, bone, liver, lungs, and eyes. All these can be donated. Some can be donated. Kidneys, skin can be donated when we are alive. Other organs only after we die. Okay. Hope this part is clear, children. In the next video, we will start with biotechnology. Now we learnt about the cytology, and we learned what are stem cells, what are the uses of stem cells, and about organ organ donation, organ and body donation we have learned. So this much is only in this video. We will continue in the next video. Thank you, children.